they own them or so. It really is upsetting. And I feel they've taken my rights away and our privacy. And we're not the only ones they've been doing too, as many others. Why are they prosecuting so many of these little farmers? Well, they're only maybe worth a couple of $100,000 when they're such a multinational company. Why? Is it greed, or is it they just want to control all the seeds?
Percy was not the only farmer accused by Monsanto.
In the summer of 2000, my family and I were discussing how rude we thought it was that Monsanto never sent us a letter or thanked us for cooperating with their investigation into our business. We were stunned when in late July of the year 2000, we received a letter from a law firm in New Orleans that was representing Monsanto Company, accusing us of infringing on their patent. Every accused grower in the United States that I have talked to had nearly identical stories. They come and test your crop. The farmers that have nothing to hide figure, well, they'll find that we didn't plant this crop that they say, and they come back over a year later and accuse you of this when the farmer can't go back and take crop samples and disprove them. Monsanto had told us that they had sampled all 3,850 acres of our soybean crop. The samplers would have had to have been gathering one sample continuously every 20 seconds. After examining the testimony that we gave, realized their story didn't add up. Now they changed their story, saying they only took a handful of samples per field. Are we to assume that they were lying in their first attempt to extort money from us, or are they lying now?
Estimates are that Monsanto has sent out 9,000 letters to farmers. Most farmers choose to pay to avoid lawsuits. Even so, there are 100 active lawsuits in the United States alone. When the farmers do settle with Monsanto, they have to agree to never discuss their settlement.
We feel that we were profiled by Monsanto because of the size of our farm, and that they wanted to try to make an example out of us to scare other farmers into never saving their own seed, to be too scared to save their own seed.
Eventually, my case went to court after 2.5 years, and Monsanto dragged me through those courts in pre-trial. They did everything to break me, and basically took all of our retirement funds, because just my lawyer fees alone up to date have been around $200,000.
One of the sons said, "Mom and Dad, I hope it won't get you down, because they're trying to destroy you. But I hope you're strong and you just get through it all." He says, that's the only way the changes will be made in our country with these big companies.
Monsanto ready to wage war. Schmeizer's case opened the door for Monsanto to pursue other patent breaches. A lot of people have asked me why I didn't just settle. I probably could have settled for maybe a slap on the wrist or maybe a few $1,000s fine. But what I did was talk it over with my wife and said, look, they destroyed our seed, everything we've worked for in the last 50 years. And if I sign this, that I grew genetic altered canola without a license when we were doing our own thing, minding our own business, I couldn't live with myself. And I feel that it was taking my fundamental rights away from me. My grandparents left a system like that way back, turn of the century, to get away from control and a feudal type system. Are we going to want to go back to that? And we said no. We'll fight it and stand up for what we believe in.
And what did the judge rule after 2.5 weeks of trial? He said it didn't matter how Monsanto's genetic altered canola got into my field. And then he went on and specified. He said whether it cross-pollinated or if it blew in by the wind, by birds, bees, animals, or falling off a farmer's truck, a combine, and so on, it didn't matter. The fact that there was some plants there I violated Monsanto's patent, even though I didn't want it in my field. Number two, which is the most important one, I think, he ruled any farmer that has a regular conventional plant, it doesn't matter what kind of plant, if it's a tree, if it's a seed, and it gets cross-pollinated with Monsanto's gene against your wishes and destroys your property, my plant becomes Monsanto's property. Now stop and think what that means to farmers all over the world, farmers, gardeners, anything to do with a life-giving form. The third issue, he ruled, the fact that I never used Monsanto's patent, which means I never used Monsanto's Roundup Ready herbicide or glyphosate on my crop, he ruled that's immaterial. He said the fact was that there were some plants there. So that shows you the power of patent law over farmers' rights.
If you go and look at a canola field that's 1/2 GMO, 1/2 regular canola, you will not be able to tell the difference between the two. They grow the same, they act the same. The only way you'll be able to discover it's theirs if you go spray Roundup. Well, what you killed is your own, and what survived belongs to Monsanto now as a result of this case.
In the last 2 months, I probably had 100 phone calls, minimum, about people finding canola in their fields that never ever had it before. And I think it would be safe to say that it would be very difficult to find one field in Western Canada that would not now be contaminated to some degree with Monsanto's GMO canola.
There's an old sort of ethic that emerged in agriculture communities, and I grew up with that ethic. And the basic ethic was that if I decided to bring an agricultural enterprise into my neighborhood that was potentially damaging to my neighbors, for example, if I decide to raise cattle, and those cattle are potentially very damaging to my neighbor's crops, it's my responsibility to fence the cattle in. It's not my neighbor's responsibility to fence them out to prevent damage to his crops. The way that the system is functioning now, it's my responsibility to fence that technology out from my crops. And of course, there's no way that you can build a fence high enough to prevent that. We know that now.
All species have developed elaborate mechanisms over millions of years to protect their cells from invasion from foreign DNA. Genetic engineers have to overcome the integrity of the species barrier to insert a new genetic trait into a plant or animal. When scientists began genetic engineering 30 years ago, they believed that one gene expressed one trait, that transgenic DNA would be stable even when forced to cross species barriers.
A new technique of molecular biology appears to have allowed us to outdo the standard events of evolution.
In 1975, a conference of pioneers in genetic engineering was held to discuss the issues of safety and ethics of the newly discovered transgenic research. Some biologists argued for a moratorium on research until the safety of the technology could be established.
It is not known what sort of risk this is going to create because we have no tests.
The conference delegates voted against the moratorium, but did pledge to keep all recombinant DNA and genetically engineered organisms contained safely within laboratories. Currently, both agriculture and medical biotechnology use genetic engineering. Medical biotechnology has created many life-saving products. These pharmaceuticals are made under controlled conditions in secure labs. Genetically modified plants, unlike drugs, reproduce, and once released into the environment, cannot be controlled.
Using biotechnology to produce food is not new. For nearly 6,000 years, people have used yeast, a living organism, to make bread rise. And we've used biotech for almost as long to ferment beer and wine.
So there's really nothing new here. But that is completely wrong. Genetic engineering is really a radical revolution in food production. It's really a cell invasion technology. You know, people have heard they're taking a flounder gene and putting it in tomato so the tomato can last in cold temperatures. But people don't ask, how does that flounder gene get in that tomato? How does it get in there? And what really happens is the only way you can do it is to invade the cell of the tomato and deposit the flounder gene. Well, what's good at invading cells? Bacteria and viruses.
After 12 years of searching, Monsanto found a soil bacteria that is naturally immune to Roundup herbicide. Their goal was to genetically engineer DNA from these bacteria into various plants. They cut out a sequence of DNA that is resistant to Roundup. But if this DNA sequence alone is inserted into a corn plant, it will have no effect. So the next step involves E. coli bacteria. Gaps are created in the E. coli DNA, and when the two test tubes are mixed together, some of the E. coli DNA recombines with the Roundup-resistant bacteria. Then the technicians smuggle the engineered DNA into the cells of the corn plant they want to modify. Cells will naturally reject foreign DNA, so they developed a method using soil bacteria that causes tumors in plants. They use this bacteria to ferry the engineered DNA into the plant's nucleus. There are also two other methods used to get the engineered DNA through the cell wall. One uses a stream of electricity to create tiny holes in the plant cells so they become vulnerable to infiltration by foreign DNA. Another is the gene gun, which blasts particles of gold coated with engineered DNA into the plant cells. Each of these three methods needs a promoter gene that turns on the desired characteristics. The promoter gene is often extracted from the cauliflower mosaic virus.
This capacity of bacteria and viruses to invade mammals in different ways is what really has a lot of people edgy about biotechnology, because that's really what the tools are all about. In order to move genetic material from one organism to another that don't normally cross, you've got to sort of behave like bacteria and viruses and invade into the cells and become established, just like a virus must become established.
And they do one more thing. They attach to that an antibiotic marker system.
The antibiotic marker is a gene which is naturally resistant to a specific antibiotic, so later they can test whether the genetic cassette is being expressed.
The most cataclysmic force in the food system right now is the fact that the medical community is just terrified about the loss of antibiotics. No one really understands how using antibiotic marker genes in genetic engineering techniques might contribute to the problem. The biomedical community on a worldwide basis is absolutely focused on this problem now.
As we move on into this so-called biotech revolution and we start producing more and more transgenic manipulations,